Okay, then we're going to start this session. Our first speaker is Fred from the Swiss Club of Bioinformatics. Please go ahead. Thanks.
It's okay, it works, yeah. Hello, so I'm Frederic Bastian. I work for the Gene Expression Database BG, developed at the Swiss Institute of Bioinformatics and University of Lausanne. And I'm going to present you today the steps we're currently undertaking to be able to integrate single-cell RNA-seq data in potentially hundreds of animal species. BG is a global core biodata resource integrating expression data from different types like Biocuration data, single-cell RNA-seq data, in so far 52 species, and our aim is to make all this gene expression information comparable between experiments and between species. And so far, we have single-cell RNA-seq data in 13 of those species. But now there is really a data deluge of single-cell atlases in a lot of non-model species, and we know they're going to be like hundreds of species with a single-cell atlas really soon available, so we want to be ready to integrate all those data. So when I say that we do integrate data between data types and experiments, I show you here the example of the Drosophila gene, olfactory receptor neuron, OR13A. And on the right here, those are the source of data. We have single-cell data, in situ hybridization data, Affymetrix data, and we do integrate all of that to provide one answer to the question, where is this gene expressed? And we identify that the top expression of that gene is in the exact corresponding olfactory receptor neuron type, the OR13A olfactory receptor neuron. And to quantify gene expression comparable between experiments, we use what we call an expression score, which is a non-parametric statistics that is batch effect free, and that then we can compare between species and experiments and data type. And it goes from 0 to 100. So here, an expression of 99.92 is really, really high expression for that gene. Okay, so now if you want to be ready to integrate single-cell atlases in other species, we have to modify a lot our current pipeline regarding dataset curations, the preparation of genomes and ontologies we integrate, how we process the data, and how we propagate all this information in what we call a graph of condition using all the ontologies we use. So anatomical ontology, cell type ontology, life cell ontology, and finally, how we're going to present this information in BG. So first, regarding the dataset curation, for single-cell data, the curation work is really changing. So we aim at capturing the information provided by the author, so standardizing the cell type information they provide. We don't aim at reanalyzing their data and rediscovering their cell type, but there is a lot of steps to extract data from H5AG files or from FASTQ, whatever. And so we decided to document all the steps we take to extract the data and to document the decision we make to map a cell population to a specific cell type term. And we use notebooks to do that. And this is a work that has been started by Saurabh, who has a poster here tomorrow. So if you're interested, come to see her. And yesterday at the single cell workshop, we loved that actually we are now building metadata about metadata, which is exactly what this is. We document how we capture metadata. So that's how we're going to create data sets so that then you can see all the information necessary. Then about genomes, we have another problem is that the gene models for non-model organisms, they are not as good as for human, mouse, fly. And here I show you a plot of to which regions in the genome the reads from RNA-seq map in the opossum genome. So each plot is one chromosome, and in orange, pink, it's intergenic regions. And you can see here that you have a lot of reads mapping to intergenic regions, which is not what we aim for with RNA-seq data. And this is really problematic with 10x technology, which is 3' end sequencing. And the 3' end of gene is really poorly annotated, mostly because of unannotated UTR regions. And so it's really problematic when we want to integrate so many data in non-model organism. Just for comparison, I showed you the equivalent plot in human, and you don't see a lot of orange peak regions because the 3' end of genes is really well annotated. So what we're going to do is that we're going to use a tool called GeneX that extend the 3' end of gene models based on single cell data so that we can increase the accuracy of read mapping from our single cell data. Then we also need to do a lot of work regarding ontology preparation. So this figure comes from the Obo Academy. I want to thank the people from the Obo Academy that the first time I see taxon constraint in UBaron so well explained. So we use UBaron and the cell ontology, and we need to infer for each species we integrate which terms exist or do not exist in those species. And for this, we use the taxon constraints that are shown on the left, so that then using a reasoner, we can infer, for instance, the example here that a catfish does not have hair because it exists only in mammals, or that a human does not have a whisker muscle because whisker does not exist in human. So this reasoning preparation we're gonna have to do for all the species we want to integrate a single cell atlas for. So our plan is to prepare this taxon constraint for all species in ensemble and ensemble meta as well. So we would be very happy if the UBaron developer would like to do it instead of us, but if they don't, we will do it. So that as soon as an atlas is available, we have the ontology and we have the genome that are already ready for the data integration. Then we have a data processing step, of course. So here I show you an example in Drosophila that comes from the fly single cell atlas, where we have data in the spermatocyte cell type in the whole organism in adulthood. And we have 7 libraries that sample this cell type, spermatocyte. And I show here expression of one gene coming from these 7 libraries in this cell population. And we have like different expression level expressed here in counts per million or UMI counts. And we have different information coming from these different libraries. But at BG, we're gonna then integrate all this information to provide one evidence of expression using our expression score, integrating all data available. So here from this 7 library, we end up computing that we have an expression score of 98 over 100, so a very high expression of that gene in the cell population. And we also provide a p-value representing how confident we are that this expression is a true signal of expression, that is not a spurious, noisy expression coming from single cell data. And here the p-value is quite low, so we are quite confident that this is a true signal of expression for that gene is in spermatocyte. So you see that we have multiple samples, and that could come from multiple experiments, but at the end, we summarize everything with one call of expression, with a non-parametric statistic of expression score, and with a p-value representing how trustful we are that this is a true expression. So once we have that, we're gonna propagate this information into a complex graph of conditions, integrating the anatomy ontology, the cell type ontology, the life stage ontology. And here I show an example in 3 species, but basically on the left it's human, on the right it's fly. So our first step, we retrieve the data and we retrieve the information provided by the authors as free text information. So the free text information about cell type was in human that this library, this cell population was oligo-Balyan llama 2, and in fly that it was enchyting glia. And so we're gonna map that to an ontology. So in human that correspond to the CL term oligodendrocyte, and in FBBT that correspond to the term enchyting no associating glial cell, something like that. So you see here that we don't have matching information between these 2 species. We cannot really compare, we cannot reconcile the information. So we're gonna propagate this information, this expression score, these p-values along the graph of conditions so that at some point, you're gonna reach a common node between these species. So at some points, you're gonna reach the node glial cell. So both the oligodendrocyte and this associated glial cell are glial cell. And at some points, you're gonna reach this node, glial cell in the brain in adulthood, that then we can make comparison between species so that you can compare the expression information in glial cell in human and fly. Then how are we gonna display this information? So I know that for single cell data, it's very common to display a dimension reduction plot as on the left, where you can see the cluster that the authors identify, and then they identify marker gene for their cluster so that they identify the cell type. This dimensional reduction plot and clustering, they require a lot of fine-tuning of the parameters. And usually authors try many different methods, many different parameters to end up with something that they agree with. And we cannot do that if we want to integrate hundreds of species. So I know that there are resources doing fully automated pipeline to render a clustering plot. I do believe this is not truthful to the work of the author. They spend a lot of time identifying their cluster and defining them. So we don't want to use a dimensional reduction and clustering plot. But what we want to do is to use tree-based methods that are a priori parameter-free, so that we can still display the cell population with distance between the cells, but remaining truthful to the author work. Okay, so in conclusion, we have several steps to update that we are updating. For curation, we are gonna document everything with notebooks. We are not gonna reanalyze the data. We trust the author information. For genomes, we are gonna prepare all genomes from ensemble and ensemble metazoa, extending the 3' end of genes. For ontology, we are gonna pre-compute all taxon constraints for all species in ensemble and ensemble metazoa. Then we're gonna provide our processing that is batch effect free with our expression score. And finally, we're gonna provide visualization that are parameter free so that we can run a fully automated pipeline on them while remaining truthful to the authors. So I thank the BG team and the photo for this work, and I thank you for your attention.
Thank you, Fred. We have time for questions. The microphone is coming.
Great talk. And I have a question. So the data for single cell data, you can browse in BG and download and do a lot of things. Are you working with any of the other data resources or knowledge-based providers? Because other resources may not have that rich way to search and visualize. Are there any distilled down version of this information that still could be interesting, but not as detailed? Then they would have to go to BG. So if you're collaborating with somebody, maybe you can give some examples.
Yeah, we organized a workshop yesterday about a consortium we started called SC Fair, where we have like a cell by gene, fly-based, Wellcome Sanger Institute on board. We would like to get also the ABI single cell expression atlas on board, and we collaborate to agree on metadata schema and to coordinate the curation work and see, we have a common H5AD format to provide the single cell information. So yeah, definitely we need to collaborate all together because there's so much work to do. We cannot do that alone anyway. So yeah, there is a website, SC Fair, if you want to check.
Yeah, a quick question.
Just a quick question. You're using notebook. Why not SDRF? I thought that's what that was written for.
Sorry, what?
SDRF.
What is it, SDRF?
The format for holding metadata.
Oh yeah, we use H5AD, you mean? Or the notebooks you probably.
Yeah, that's why are you using notebooks and not SDRF, which I thought was the agreed format for RNA-seq data.
So it's not about RNA-seq data, it's about documenting how we annotate the RNA-seq data. So we could do that with any data. It's not related to single cell, just our way to provide additional information about the curation process. So it's totally unrelated. And notebook, they can be used to document anything, basically, any analysis.
Yeah, you're still doing an SDRF file as well?
I'm not familiar with SDRF file, actually. Yeah, sorry.
Okay, thank you very much. A second round of applause to Fred.
Yep.
Talk about annotations in protein resource. Thanks.
Hi, I'm Randi. Thank you very much for this opportunity. I'm not sure if I should touch this. I'm going to talk about how we added some epitope annotations to some very well-known protein resources through a collaboration. These 4 different resources collaborated together. I work at the Immune Epitope Database, and we recently created the Cancer Epitope Database for cancer data. And these are free public resources. And the IDB has more than 1.6 million antibody and T-cell epitopes that were derived from the literature through manual curation, extensive manual curation that relates to infectious diseases, allergy, autoimmunity, and transplantation. And the CEDAR database is specifically cancer data. And I think everyone's familiar with Uniprot KB. We worked with them, the database protein sequences with all their rich annotations, and the protein ontology, which has the ontological representation of proteins with explicit proteoform information, and IPTMNet, which is a database of post-translational modifications on proteins, and they integrate this information in a systems biology context. So this is the homepage of the IDB, and the only thing I wanted to point out, let's see if this works, is the red box, that's our metrics to show that we have people, PhDs, who have read more than 25,600 publications, and they extracted all the experimental details that relate to an immune response in those papers where the epitope is known. And I know most people don't know what epitope is. So the epitope is this small portion of a protein or a non-peptidic structure like a chemical or a drug carbohydrate that the immune system actually recognizes. Antibodies and T cells don't recognize whole proteins, they recognize a small part of it. And so the majority of the data in the IDB are peptides from proteins, and that's the link between these other resources in the IDB, as we've manually curated a lot of information out of literature about proteins, and we want to share this with protein resources who are the experts in proteins, and we are definitely not protein experts ourselves, we're epitope experts. So on the IDB, we have the antigens, and so each antigen for us is mostly proteins, but it could be a large lipid, for example. But so for each protein, we have a summary of all the information we've curated. So hemagglutinin from influenza is the most studied protein in the field of adaptive immunity because it mutates every single year, and you need to get a new flu shot because your immune system doesn't recognize it after it's mutated because those epitopes have changed. So we have in our database curated more than 794 papers with describing more than 3,500 epitopes from that one protein. So for each protein, we have accumulated all this information, and we already have it. That's our mission to do it. So we wanted to share that with these protein resources. So when we curate, we have to aggregate all the data from all the different publications that describe the same protein. So that same hemagglutinin is actually hundreds of different hemagglutinins. So each of our curators have to accurately and consistently capture this data. And then when we put it on our public website, we have to efficiently and intuitively present it back. And those are two very different problems and have different needs. So we employ different strategies at the curation and the collation, and then now through this collaboration to accomplish those goals. So for curation, our curators need all possible proteins available to them. We even make up our own sometimes when they don't exist, especially for more rare species where there isn't enough information deposited yet. So this is examples of 3 different publications with the PubMed IDs in green, and which hemagglutinin the different curator chose to represent that epitope. So the first epitope is discontinuous residues. Antibodies typically recognize specific residues, not something linear. And so that specific GenBank record contains exactly those amino acids at exactly those positions. And it has to be true to the paper and what the authors discovered. That protein sequence has to be 100% identical with the epitope sequence. So our curators use anything, whether it's Uniprot or GenPept or PDB or whatever is available. And because there is some data for species where not a lot of proteins have been deposited, sometimes we have to make up our own, which we hate to do because, again, we're not protein people. And we periodically go back and see if new, better ones have come out. And then we collate this data using Uniprot reference proteomes. So for each species, we pick the one proteome that exists at one point in time that best represents our data. And we do that through the sequence identity of all the data we have up until that point, the day we're picking the proteome. And we periodically check back because new proteomes became available. And also the data changes over time, because people start studying something different and our epitope sequences may shift, and then a different proteome may better represent. So for influenza, we've been using this APRH proteome for a long time, and viruses are great for examples because people are familiar with them, but also because they only have 13 proteins in the proteome, which makes it nice and compact. So what we do on the IDB is we display that back to our users as that list of proteins per species. We do it on a per species basis. So when our users are searching, they just see one hemagglutinin and they click it, and then they get all the data for that protein. And then because we use Uniprot, we get all of the synonyms that Uniprot has because we rely on them to be the protein experts and to find all the different spellings and abbreviations. because a lot of people talk about proteins as if they have one name, and it's absolutely not true. And especially in the field of cytokines, the names have changed over the years, and some of them are completely different, and you would never know they were the same unless you were really old and familiar with the history of the literature. So through this collaboration, we added new features to the IDB and new features to those other websites. One of the first thing we added for the IDB was a new antigen summary pages, which summarizes everything we know about that protein that we've gotten from the literature. And because we started from the beginning of PubMed and we stay up to date, this is everything that's known about the adaptive immune response to this protein. These are all the species in which it's been tested. These are all the disease contexts in which there was an experiment and a publication. And so because we've already pulled this information out, we want to share it with the world. Even if it's just specifically about an adaptive immune response, it still was very labor intensive, and every bit of data links back to the paper it came from, so the other resources can decide for themselves if that's interesting or useful. So using Uniprot, in the past, we just took the reference proteomes, but now through this collaboration, we're looking more into all of those rich annotations and being able to add more information to our website now that we have those antigen pages. So we're adding gene information, other function information that we just didn't bother with before because we didn't have the page. And it is useful for our users. So it's nice to take the time to look at the data and see what else we could improve on. And then on those antigen pages, we also link out to PRO and IPTMNet. And this is just a particular protein that has been studied in a lot of cancers. And with the immune system, it's very common for an immune response to be to something that's not modified or only is modified. So it may not recognize the unmodified peptide, but it will recognize the modified. So post-translational modifications are important to immunologists. So if you click on the IPTMNet link out, you get all the information about all the possible post-translational modifications for that protein. And this is new information we're providing to our users who may never have heard of IPTNet and didn't realize this resource was out there, which is the goal of like sharing our different goals. And then Uniprot added a new feature viewer for epitopes to their page. So along the links to the protein, you can see now where there are the most epitopes concentrated. And it's actually really common for the immune system to recognize specific parts of a protein very highly, in other regions not at all, just based on the sequence. And then if you click on the epitopes, the little green bars, you get a pop-up that links back to the IDB, says where the information came up from, and says the weight of that information, how many experiments there are, what type of experiments they were, and exactly what paper they came from. And then Uniprot also used all of our disease information, and they've added new annotations to describe the disease context in which those proteins have been studied. And that's actually really interesting for cancer, and especially autoimmunity, where it's common. So this human myelin basic protein is important in MS and neuromyelitis optica. So it's common for the same proteins to be implicated in various diseases. And then you can also see the weight of how many papers have described that. And then PRO added more than 20,000 proteoforms that relate to epitopes in the IDB. And so they link, so each of those red IDs links back to a page on the IDB for an epitope and gives you the information about that specific proteoform. And then the main reason we decided to do a talk is that whenever we all work together, lots of good benefit comes to all sides. So besides those features were all new things added to the web pages, but we also fixed problems, and that's my specific favorite thing to do at the IDB is to fix problems because they're hard to find once you have a lot of data. So by doing this analysis, PRO helped us identify, Uniprot helped us identify obsoleted protein sources that we were using that we would have never noticed they were obsolete. And so then we fixed those. We found cases where a curator had mistakenly called a protein the wrong species. So they may have taken a human protein and then called it cow. We found cases where post-translational modifications couldn't be possible. So either the curator referred to an amino acid that didn't exist in that protein sequence or in a place it didn't exist. And so we fixed all these errors and we added validation to prevent them from happening again. So going forward, we want to continue and add information about cancer antigens now that we have the CEDAR database, autoantigens and allergens, We can give weight-based evidence based on how many papers and how many experiments describe the same phenomenon. And we could expand our antigen summaries to include non-peptidic structures using KBIDs. And this is a collaboration between a lot of people. So I want to give credit to everyone at all of these sources because everyone took a good amount of time to look at how we could overlap. And hopefully they can continue to find other overlaps, and that's why we come.
Any questions from the audience? Oh, yeah.
Hi, I'm Mark, Reactome. You sort of zip by this. No, I don't mean it like that. I mean about choosing a strain and then as the sort of progenitor for all the proteome parts to be attached to. And we find that really hard, actually. So many. Are you interfacing with like experts working with those viruses and that's how you choose, or is it just a best fit? How's that work?
So for one protein, we take every epitope we have data for, and then we blast match it to the proteomes. So we'll check every human gluten and see which, and then we have to take it as a whole too. So humans have so many proteins. We do a procedure, I think 85% identity is the minimum to consider, and then we just take the one proteome that has the best sequence identity to all of our data for that species.
Still have time for a quick question? Really quick, please.
So when in the paper, the peptide is different than the reference,
Yes.
and how much do you track that and how difficult it is to keep track of that?
We do track that because it's something that can be confusing for our users. Sometimes, when we provide the data back, we provide two sources, the 100% matching source and then the reference proteome source. And they can use them both for some people only care about that one representative and they don't care that it's not 100%. But if you're looking at the immune response, you make an immune response to this one and not that one. So it can be very important depending on the type we have a lot of different types of researchers. So we give both. We try and be very clear about it because it is a point of confusion. Okay.
Thank you all. And let's give a second round of applause to Randi. Thank you, Randi. Our next speaker is Sonia. She'll talk about the Phenome.
So a very good evening, everyone, and a warm namaste from India. So I'll be talking about the initiative of Indian Biological Data Center towards making Phenome fair. So before going into the details of Indian Crop Phenome Database, I would like to introduce you to the Indian Biological Data Center as we are the new repository. IBDC is the national repository for the life science data in India. It is funded by Government of India through the Department of Biotechnology, and it enables the implementation of the Biotech Pride Guidelines, which is formulated by the Indian research community. It's a joint project of Regional Center for Biotechnology, National Institute of Immunology, and International Center for Genetic Engineering and Biotechnology, and also the National Informatics Center. We have a disaster recovery site at NIC Bhubaneswar. This is just to give you a brief overview about the campus. We are with the two national importance institutes, like we are under the Regional Center for Biotechnology, and we have a neighboring institute like Translational Health Science and Technology Institution. So what exactly we do? We, as a new repository, we try to provide the research community with the biological data solutions that we, for different types of data. We provide high computational facility to the research community. We support our national projects of national projects like INSACOG and Genome India. We do big data analytics. We provide users with different resources. We do a lot of training webinars and help users with the submissions and the data analysis. So coming to the what all portals we have at IBDC, we have started with nucleotide data archive portal. So we have these two portals, INDA and INDA-CA. Now we have a total of 7 portals that are functional, IPD for proteomics, ISDA for structural data, metabolomics. We have IMDA, IBIA for the image data archival, and ICPD for the crop phenome database. We are also working on now expanding the phenome archival to Indian human phenome database and Indian animal phenome database. Also, we are working on developing some tools for the multiomics analysis for the research community and for the GWAS data also. This is again another repository on which we are working on to sustainably archive the other data which is associated with the research. So coming to the phenome data, as we all know that these are the set of methodologies and the protocols that are usually required to measure the plant growth, architecture, and compositions. It can be more in traditional way the phenotyping has been done, but now we are going forward towards the high throughput phenotyping, which we often call as phenomics. But as per the latest survey, as you can see, as you can see here in this map, like the condition of India, where we mostly rely on the traditional phenotyping methods, and recently the high throughput phenotyping has been introduced in some of our institutions. So it's a mix of different methodologies that our research community follows. So, but agriculture, as per, it is very important for India, as most of it is the biggest employer in India, and majority of the population still dependent on the agriculture for their livelihood. It also contributes a good amount of proportion towards the GDP, and again, being the most populous country in the world, we need to take care of the food security as well. And then our government is doing efforts towards the allocation of budgets towards this also. But as a researcher, what exactly this data is hold importance, because this phenome data with all of the detailed metadata and another omics data, GWAS data, QTL can hold a very diverse applications where food security, agriculture, precision, data-driven decision making, and bridging the genome phenome gap, engineering different cultivars which are climate resilient and also high yielding varieties. So, but the challenge here as Indian ecosystem that we have is the data we generate is very heterogeneous, and it's very difficult, and it is never managed as per nationally. So every lab has their own data, then it got lost in the labs as the student goes, and it is very complicated process to observe this, because of many types of data it can involve, and it has never been assembled by humans and machines. It is very poorly documented and very rarely reused. So the fair data cycle is missing as we compare the phenome data with that of the other omics type of data. So with Indian crop phenome database, we are initiating a process to structure this agriculture data that is generated in Indian labs, and we provide persistent IBDC accessions. I will try to make it a central portal for the submission, storage, and access of crop phenotype data. We use ontology-based, very simplified user-friendly interface, and currently we are giving 2 access options for the users: open and managed access. In a way, we are trying to support the fair and sustainable phenotype data. So we try to collect all the important metadata fields from the users as listed here. And this is the homepage where many of the tools and user resources, webinars were there for the users. The inspiration is again from MyAPI, NG, and PIS, which are the international standards available for the phenotype data. These are the basic schema where user can submit many of the projects. One project can have multiple studies. Then we have a genotype samples data file, which have all the observations. And then we have a whole lot of metadata fields and associated data, also the crop passport data together. So in a way, the metadata is like we try to give users a framework where if he has single project, he can able to submit multiple projects. It can support multiple crops per project. It can support collaborations, and different types of schemas are there. So whenever a submitter submits, he will get accessions for project, studies, and data files. In a way, he submit, data is archived, ICPD accessions were granted, and the data is accessible to the research community. Then we use crop ontology and plantiome ontologies to enable the data submission platform. These are the detailed metadata fields that we collect during the submission process. And then this is the simplified version of the process, that for every submission, user needs to create an account, log into the dashboard. He can create the Phenome project, register as many studies, and then each study will have a data file, and then it is submitted and accessions were granted, and now it can be accessed by the users. Then this is the format of the data file, that this is template-based submission. And as soon as the user enter all the metadata, this automatically generated template would be there. And he has to only provide the observation values and upload it back to the system. So in a way, he can also use these files for documenting data. And then in a way, a structured way of managing his data can be followed by the students as well as the research community. This is the dashboard of the ICPD where these are the test data. So really don't try to meaning out of that. But all these fields can be there. The dashboard will show how many projects, how many studies, how many data files are there. And also he has the access to different tools, like he can browse ontology. If the ontology terms were not there, we will provide him the facility to create ontologies for method, tissue, traits, etc. The manual is there. Then in a way, this is a complete dashboard in which all the phenotype data can be managed. And this is the current data statistics. We are getting submissions now as the research community has been motivated by the funding agencies that they need to submit, otherwise the funding agencies will not fund the project further. So they have realized that this data needs to be structured in a way. So we have users from different institutions in India. As of now, 26 projects, 196 full data submissions were there. 7 crops were, you can find 7 crops data in that. We also have data for the major mission mode projects on rice, wheat, safflower, and linseed. Then you can see that majority of the data is from wheat, and followed by rice and sesame. Then these are the different meta traits that has been submitted by the users. These are the number of trait which has been submitted, number of tissues, methods, and developmental stages. As of now, majority of the data is on biotic, and some are in controlled conditions also. So these are the genotypic samples per study. Then these are the ontology-based systems. So we provide users with the browse ontology and create ontology. So every ontology terms he create will get in temporary accessions. And as of today, we have these blue bars shows that these ontologies were created by user, which needs to be curated and then submitted to crop ontology. So this is the browse page where all the studies can be found based on whether the user choose controlled access or the open access. So after complete submission, these studies can be easily accessed and data can be downloaded, easily shared. So in future, going forward with the future, we want to analyze this data in light of the other data that we have in different portals, because many of the mission mode projects, we already have genome sequencing data in the another portal. So we wish to create a resource that is specific to different crops. And this is the woman-led team of ICPD. Myself, Nivedita, Isha are data curators, Mayuri is our software developer. Now we are expanding to the integrated Phenome repository of India, where Vibha is also helping us. And previously, Pratyush and Abhay also contributed towards that, and Professor Saurabh Raghuvanshi as our former lead also guided us in this project. So these are the PIs of the IBDC, and this is the whole IBDC teams. And we acknowledge the funding from Department of Biotechnology and our partnering institute. And and last, I thank my 3 kids because they allowed me to come here, and this is for the first time I'm away from them for such a long time. So thank you, everyone. Thank you so much.
Thank you very much, Sonia. And we have still time for a quick question. There.
This was super interesting. Looking forward to exploring more. I see that you guys are looking into some tangential areas. I'm wondering if you guys are looking into or thinking about looking into water as well.
Water, yes, definitely include that. We have the functionality where user can provide any information which is necessary for his study. So he can submit it as associated data. So any data which is essential for the study can be uploaded together. Thank you.
Thank you very much. Thanks, Sonia again. Our next speaker is Nicole. Okay.
Thank you. My microphone's working. Yeah. Hi, I'm Nicole Vasilevsky. That's a great talk, Sonia. I'm going to talk about something similar in a way, except for a database for sharing rare disease, human rare disease data at the Critical Path Institute called the Rare Disease Cures Accelerator and Data Analytics Platform. So yes, I'm Nicole Vasilevsky. I'm the Associate Director of Data Science at the Critical Path Institute. This is my cat, Frankie, who helps me work. And so the Critical Path Institute is a biomedical nonprofit that's based in Tucson, Arizona. I work remotely in Portland, Oregon, but I definitely enjoy going down to Tucson and seeing the saguaro trees and all the beautiful mountains there. So our institute, we're funded by the FDA, and we're a pre-competitive neutral convener between industry and academia and others and the FDA. And our goal and our mission is to advance drug development in underserved disease areas, including rare diseases. So if you're not familiar with rare diseases, the definition changes by the region that you're in, by country. In the United States, it's defined as any disease or disorder, which is also controversial what that is, how to define that. But it's a disease or disorder that affects less than 200,000 people in the United States. And like I said, the definition varies depending on the country or the region that you're in. But in the US, maybe 1 in 10 people are affected by rare diseases. And the majority of these cases have an underlying genetic cause. So in the cases of rare diseases, there's a lot of challenges with getting treatments into these patients and also with the diagnostic odyssey. So for rare disease patients, a lot of them undergo this long path to getting a diagnosis where it can often take somewhere between, on average, 4 to 5 years to get a diagnosis. Sometimes it can even be decades. And one of the other challenges with developing drugs for rare diseases or repurposing drugs is the small populations of people who are affected by these diseases. So oftentimes there's not enough patients to create a cohort for clinical trials, making it difficult to carry out trials and advance treatments and discovery of new drugs. And like we've seen in a lot of different cases, data is collected from rare disease patients, but often can be heterogeneous or siloed or disparate. So we need to be able to aggregate this data and break these data silos. So using standards like we do in Biocuration to standardize the data and harmonize it can help to maximize the utility of this rare disease data. So at CPATH, one of the big projects that we're working on is called the Rare Disease Cures Accelerator-Data and Analytics Platform, or the RDCA DAP. And you can go to our portal. There's a URL at the bottom of the screen where you can go to our portal. So we are taking many different types of data, including clinical trial data, data from patient registries, like specific rare disease registries, natural history studies, genomic data. We're just setting up infrastructure now to ingest imaging data and other types of data, including preclinical data. We are aggregating it into our platform. At CPATH, we have a dedicated, we call them data managers, a data management team who are the ones who are responsible for collecting the data and standardizing it to different standards. So the data is shared with us via, we have data contribution agreements that we work with the data providers to share this data. And then our data management team ingests the data, carries it to different standards. And the team that I'm on, the data science team, we are working on building a knowledge graph that we can integrate the data into and to be able to find novel connections between the data. And then the data is also shared on our platform called the RDCA DAP, where you can log in, request access to the data, you can browse through the data, be able to select data sets, and then we have a workspace where we can load data sets into, and there's analysis tools that are available, or you can bring your own tooling. But the ultimate goal of this platform is to be able to provide actionable rare disease drug development solutions and help advance discovery or reuse of drugs for these types of patients. So when I joined CPATH, I've been there for 2 years now. Previously, it was part of the Monarch, or still am part of the Monarch initiative. We collaborate very closely. But when I joined, the kind of data curation processes had already been kind of set up, and we're still kind of trying to figure out the best way to curate especially these disparate types of data types that we're using. There's not really any kind of one size fits all standard that fits everything. We do a lot of curation using OMOP for a lot of the real world data. A lot of the survey data, we're using OMOP as well. We've also been leveraging Tiffany Callahan's OMOP to OBO pipeline to convert OMOP data into OBO ontologies. We've also been using direct mappings with Mondo disease ontology, which Sabrina talked about earlier, and I'm also involved with. We use the human phenotype ontology for annotating human disease phenotype, the phenotypes encountered in these diseases. And all the data to the clinical trial data standardized to SDTM, the study data tabulation model, which is the standard for clinical trial data. So we are integrating this data. We are leveraging the Monarch knowledge graph, which Sabrina had showed earlier, that connects disease information to phenotypes and anatomical entities and gene information. We're taking our patient data and integrating it into this knowledge graph so we can show these connections between this information that we are gathering from our different sources. So one major effort that we've been working on recently in collaboration with Mondo, and Sabrina Toro was also involved in this effort, is an effort to work with our community to help improve some of the standards that we're using. So we're working with the Mondo disease ontology to represent the information that the diseases that we're using. We have different collections of data in the RDCA DAP, and one of them is focusing on rare epilepsies or rare diseases that have kind of epileptic or seizure-like features. So we went to, in Mondo, we had a particular effort where we focused on the reclassification of the epilepsy branch in the Mondo ontology. So we held a series of workshops over several months where we brought together, we had data contributors or experts who worked in the epilepsy field from various different backgrounds. We had geneticists and metabolomics experts and epileptologists from different perspectives. We had an effort where we wanted to try to align better with one of the community standards called the International League Against Epilepsy. So we had these virtual workshops that we held over, I think it ended up being about 6 hours in total of time, where we came together and talked about how to best classify the different disease types related to epilepsy and align with this current publication from the International League Against Epilepsy. So it was super successful. It was a really productive effort that we did. Surprisingly to me, there were over 300 epilepsy terms in the Mondo disease ontology. And I was personally not aware that there were so many different subtypes of epilepsy, but it's a very complicated disease as it turns out. And we sat down and reviewed specific branches and sub-branches of the epilepsy ontology with these experts and had a lot of conversations and discussions about how to properly classify these. There was a lot of different approaches that people preferred. Like Sabrina mentioned, we have people with different preferences for disease naming. We also have different preferences for how to classify this information. Like there were specific conversations around how do we represent genetic epilepsies or how do we represent immune epilepsies and such. So this was a very successful effort. We ended up adding 21 new terms, mostly grouping terms that kind of group together different branches of the ontology, a lot of revisions to synonyms, some merging of terms and obsolescence. And this work is still underway. We're still working with other members of the community to implement some other things, such as how do we define epilepsy syndrome? A lot of these disease definitions end up being very tricky. But this was a great effort that we had, and we're gonna, in the Mondo ontology, we're gonna apply this to other areas of the ontology too. This kind of virtual workshop gathering together the experts in the community to try to agree upon how to best represent these standards. So yeah, the epilepsy data sets and other data sets are available on our platform. And like I said, this is the URL below. You can go in and sign up for a free account and browse the data. And the availability of the data, like Sonia's database, it depends on the contributor, whether it's fully open or if there might be some restrictions, how the data is accessed. And so it's controlled at the level of the data contributors' preferences, but everyone can go in and look at our what we have available. And we're continuing to collect new data types and thinking about how to best curate and standardize these. We, like I said, we're working with a lot of different standards, and it's really been kind of a fun challenge trying to think about how do we best standardize these different types of information. We're trying to apply different AI methods to accelerate some of these processes too. So it's great to be here to hear about all the efforts that everyone's working on. And just to acknowledge that we're funded by the FDA, and we're very grateful for this opportunity to bring together this data. And thanks very much to our team. The top 3 rows are our data science team at CPATH. And then, like I said, we've been collaborating with the Monarch Initiative. Nico Matanzulu and Kevin Shaper have helped us a lot with our knowledge graph development as well. Thank you.
Really on time, sharp. Have enough time for several questions. Please, yeah.
So you work with experts. Do you also work with patient advocacy groups?
Yeah.
And can you tell us like how that works out in your curation workflow or?
Yeah, we do have patient advocacy groups that are involved. Some of them help with it. We actually have one in particular who was involved in the epilepsy workshop, who is actually, she's a scientist and a patient advocate. She started the LGS, the Lennox-Gastaut syndrome. So yeah, we've been getting input from her. Having her in particular, having the scientific background has been extremely helpful, but there are, CPATH in general, they do reach out a lot to patient advocates, and they're involved in a lot of our processes. So to get that input from the community in that way is super helpful. But with regards to like ontology development, we just try to involve them as much as they're interested and want to contribute. We welcome that.
Still have time for one or more, 2 questions.
All right, technically this is not me. This is from Bridget Meadow online. So this is says Nicole, great talk. What other aspects of the disease did you bring into the epilepsy ontology? Genotypes, phenotypes, clinical indications, etc. We are modeling something similar through the EBI Industry Partnership Program.
Oh, nice. Well, this was specifically for Mondo, so we were just basically defining the disease entities themselves and the classification. There was a lot of interest, especially from some folks in particular, about modeling the gene-related features of the diseases. So there's been a lot of interest in Mondo of adding kind of the diseases as like a specific gene-related type of epilepsy, but adding additional features is kind of out of scope for Mondo. We usually just model that within like the data model and the knowledge graph itself. So, but Bridget, I'd love to talk to you further about this if you want to talk later.
Okay, then if no more questions, then let's do a second round of
Yeah.
So, Nicole, thank you. And our next speaker, Saurabh, I don't know if I'm pronouncing correctly.
Perfect,
Saurabh,
perfect,
okay.
perfect.
Nice, okay. Then
Thank you. Thank you. Very good afternoon to all of you. And I would like to thank the organizers for giving me this opportunity. It's a wonderful institute and the weather is even wonderful. The trees are so pretty. So today I'm going to talk about a perspective more than the data, because this is something that we as a group are kind of started just a couple of months back. But I still thought it's a good idea to discuss about it because we want to extend our expertise and interest into something that we were not doing earlier. So the title of the project is itself interesting in the sense that many of you may not be able to figure out what is Project Shail and what does the word Shail mean and what does curating a mountain mean. So let me explain that. I'll start with a little introduction about me and my institute. I work as a professor in the Department of Plant Molecular Biology in Delhi University. So Delhi University is one of the oldest institutions in India, more than 100 years old. And we have 90 colleges affiliated with the university with a staggering amount of students. That's about 700,000 students affiliated throughout the different colleges of the university. So it's a huge institution. We are a very small part of it, rather one of the smallest departments probably. But we do good work. We do a lot of work on genomics. So that's what we are trying to do. We work on genomics. We work on small RNA mediated, lot on small RNA mediated stress, metabolic stress pathways, regulatory pathways, especially on microRNA. We developed some data digitization, semantic data digitization techniques also. But that's not what I'm going to talk about. We were lucky to host ISB, the last year's ISB at Faridabad in collaboration with our department and IBDC. And that was very exciting, and probably we can do it a couple of years late also. Next one. Now, this paper actually, this I found very recently in the latest publication of Cell. And this basically summarizes what we are trying to do. And it shows the importance of the plant metabolites in human health. Now, what it shows is that the plant metabolites are, when they move into the gut, the microbiome processes them into further smaller molecules, which have a very profound impact on the gut health, including antimicrobial activities. So that basically means that, and we all know that, the plant metabolites have a huge medicinal impact. And this is an interesting fact, which I found that almost 30 or 35% of the FDA-approved drugs find some lineage into plant-derived products. And that's very interesting because why it is interesting for me, because I'm a plant biologist, that's one. But then as a country, we have a huge amount of traditional knowledge which is connected to that plant biodiversity. And that's what this project is all about. So about the project, the name Shail actually is a word for mountain in Hindi. And the mountain that we are talking about is Aravalli. So this Aravalli range is about 670 kilometers long. And Aravalli word itself means a chain of mountains. This mountain range is about 2 billion years old. It is older than the Himalayas. And it is very exciting because
see the location of that mountain range in that figure. It is actually spanning one of the most populous states in India, and it ends right about 1 kilometer close to our university. So it is right there in the national capital region. And it is one of the oldest mountain ranges of the world. Now, interestingly, if you look at that map over here, it on the west side of this mountain range is the entire Rajasthan area, which is basically a desert island, desert state. And so these mountain ranges play a very critical role in actually differentiating, actually demarcating the end of the desert, and they actually hold that kind of hot airs to the coming to the western eastern region. They have been a cradle of civilization to the extent that older major kingdoms as well as tribal ancient tribes of India actually, which have a history of about, let us say, thousands of years, maybe 2,000 or 3,000 of years, they have a continuous inhabitation in that area. Now, why this is important is because two things get represented over here. One is biodiversity. It is extremely a cradle of biodiversification, both animal and plant biodiversity. And then it couples with the traditional knowledge which people have on the usage of that biodiversity. And most of it is in medicinal usage. The knowledge that the tribal people have is transmitted most of the time orally. Only now attempts are being made to put it on traditional literature kind of review. But the problem comes that the way they identify a particular plant, to the extent that the name of the plant itself is going to be very different by the name which we recognize. Even I can't actually recognize it. So if we want to get that information in the mainstream, we need to have several levels of curation before that plant can actually be used up for any biomedical research. So another aspect is there are a lot of monographs which have been written on this, but none of them actually provide information where you can actually confirm or analyze the biological activity of that plant extract. It will tell you that this plant extract is good for this disease, but why and what aspect of that disease, that is not known, because this is something that was figured out thousands of years back. So we are trying to fill up that lacuna. We are trying to build up a knowledge base where we can pull in all the labs who are working in biodiversity as well as drug discovery and disease biology to use this kind of a data. Now, besides the knowledge of the tribals, we have very well-established traditional medicinal systems like Ayurveda, yoga, Unani, Siddha, homeopathy, and naturopathy. And yoga, people somewhere, all of us are aware of, but what many of us may not be aware of is that yoga is not just exercise. There are a lot of essential oils and medicinal oils which are used in massages. They are used for treatment. The interesting thing about all of this is that they may all be using the same plant or the same concoction of extracts, but their preparation is totally different. For example, tribals mostly use aqueous extract and fresh extract of a plant. Whereas in homeopathy, it is most of the time are alcoholic extracts. Now, what is happening over there is that when we try to make sense out of it, what happens is for a single plant, we end up actually having a lot of different ways where it can be used. And we try to categorize a lot of medicinal formulations, and some of them are so complex that they have components from 50 different plants. Which tissue, maybe leaves, maybe roots, that is something that is very, very varied. And that is what we are trying to catalog, and it's kind of a mind-boggling for us. Nevertheless, there is a need, which we understand, there is a need for many different groups to come up and study this. This is another example where actually this is what people have done in US at National Cancer Institute, and find that very interesting, where they have generated a metabolite repository for almost more than 230,000 extracts. Now, why they have done that and why we are also wanting to do that is that labs which are involved in medicinal biology or which are involved in drug discovery may not find it convenient to go to the forest and get the extract, because getting the extract itself is a very tedious process and you don't have these plants cultivated in the area. So they are preparing a metabolite repository. But what you see over there is that the green dots in the Indian map, they are very, very less. That means most of the biodiversity in India is not getting characterized in that manner. So that's why we need to take this thing out. Now, we looked up at the Aravalli region and the literature, and we were able to, within that, we were able to find at least 300 different medicinal plants, trees, shrubs, creepers, which are recorded to have some medicinal property. And the thing is that their description is very, very varied in the sense that which part of that plant is being used and what disease it is connected to, that is very, very disorganized. So we started to map disease ontologies to figure out, okay, this is the disease that this plant is associated with, and this is the plant organ that is being used to get that extract to make that formulation. So we're hoping that we would be able to at least put some structuring into that data. Now, the number of diseases that we were able to find for those 300 medicinal plants is also, I find this very staggering. We have infectious diseases, non-communicable skin diseases, respiratory. I'm not a medicine person, but there's a lot of variety of variability in the type of diseases that can be handled over here. And one of the examples is Boswellia, which is involved in osteoporosis as well as arthritic treatment. When we look at the genomic data, interestingly, out of 300, only 13 to 14% of the plants had any genome data available for them. And for that also, the data was not to the level that, my point being that the Earth Biogenome project is kind of an iconic in the sense that all genomic data must correspond to that kind of formats. So it's a short read data and it's not very well annotated. So that is also something that we are handling over here. So in totality, if you look at what we are trying to do, I'm not putting up much of the data today, but what we are trying to do is we are trying to curate this entire system so that we pick up plant by plant, we generate genomic data. On the side, we also generate a lot of literature and curate a lot of literature data. We are generating transcriptomic as well as metabolomic data and also ionomic data, because in many places we have seen that when medicinal extracts are described, they are described in terms of metabolites which are there. But people miss out the ions which are present during the extraction process, which may play a very important role in the efficacy of the extract. So we are generating that also, and also the soil microbiome data, because if you grow these medicinal plants away from their actual natural habitat, their metabolites actually change. And the soil metabolite and microorganisms decide that to a large extent. And it is also very important because this Aravalli Hills is very rich in copper and zinc. So there have been extensive mining. So there is reforestation which is being done. But that reforestation, the soil microbiome is not the same as it was probably 100 of years back. So we don't expect that the metabolome will also be same in that. So with that, I'll conclude, and this is my collaborators. We have Dr. Saloni Mathur from National Institute for Plant Genome Research. She's an expert in genomics. And Dr. Shail Vyas, he has a biotech company, Biohaven 360. Myself and Professor Sunil Purohit, who is a professor in botany, retired now, but has an extensive knowledge in that area. He resides in that area, and he has connects with actual tribal regions. So that's why we are he's helping us in getting the information directly from the people. So this is something very challenging, and I find it right to discuss it before even we get the data. So, if there are any questions, I'd be happy to take. Thank you.
A question over there?
Hi, thanks for that nice talk. I'm curious how you think that your research will align with other different kinds of traditional medicine and other cultures that have similar kind of pharmacopoeia, where people are also interested in understanding the
So that's one question that we are trying to address. How will it align? Because when you try to align these things, there's a Chinese medicine. There's actually in every ancient civilization, you have something of that sort. But when you try to compare it, the depth in which these studies are detailed out, that's not good enough. So that's what we are trying to do, so that if we do it in this precise format, we won't be able to do it for all the 300, but we will figure out a format or a manner in which the data should be presented so that it becomes compatible in other systems also, right? That's the idea. Thank you.
One more quick and last question, please.
So at the end, you mentioned gathering additional data from the native populace of the mountain itself.
Yeah.
So the challenge of that seems that you're combining sort of traditional quantitative approaches with qualitative approaches with interview kind of data. Are you trying to integrate that into your pipeline, the analysis of this, or do you treat these interviews as if they're manuscripts?
So actually, if I talk about India, there is a lot of knowledge which goes by, let's say, experience, that we have that community which knows that. Now, I don't know whether that is effective or not, right? And the only way to do that is to put that conversation in link with publication, if there is. Even if there is not, then there is a wide chance that people can pick it up and check it out. Because as somebody in the talk earlier said, that only 1% of this entire discovery space of plant-based medicine has been done. There's a whole lot of scope. And we need some orientation into for a particular disease as to which plant to pick. So I just want to direct them to that plant and then probably build up a metabolite repository so that they don't have to actually go to the forest. They can actually order the metabolites and start their work. So it's drug discovery, as I understand, is a 1 in 1,000 chance. So based on that, I need to enable people to get that 1,000 instances. Otherwise, you pick a plant, you try to find a drug, you do not succeed, and you end up concluding that it's not good enough. So that's what we are trying to solve. It may not be successful all the time, but then we would answer it for final. Okay, this doesn't work. That's the thing. Yeah.
Thanks a lot, Saurabh. And,
Thank you.
Our next speaker, Stan, talk about the Rat Genome Database.
Okay, my name is Stan Laulederkind, and I'm a curator for the Rat Genome Database. And I'll be talking about one of our latest projects, which is the import of human GWAS data and mapping of EFO to multiple ontologies at RGD. Now, the first thing you should notice about the title is that RGD isn't just about rat data. It's also about human data. We import human data, and we also do manual human data for disease and phenotype. And since one of the main needs of a model organism database is to connect preclinical data with human data, we wanted to import GWAS SNPs from the from the NIH EBI, a human GWAS catalog. So we imported thousands of SNPs from the catalog, but the annotations are done in EFO, the experimental factor ontology. So the problem was that we don't use EFO for any other data at RGD. So we needed to map EFO to some other ontologies that we do use at RGD for phenotype and disease data. Okay, so this is a list of the various ontologies we use for disease and phenotype at RGD, depending upon what type of data it is. So on the top line, that's qualitative rat data, rat phenotype data, and we use the mammalian phenotype ontology, MP. For quantitative rat phenotype data, which is a process we use called Phenominer, there are numbers of different ontologies that we use, including vertebrate trait ontology, VT, clinical measurement ontology, CMO, measurement method ontology, MMO, experimental condition ontology, XCO, and UBRON, and the cell ontology. For human phenotype data, we use the human phenotype ontology. So that would be our manual annotations, the human genes for phenotype. And the last on the list is qualitative disease annotations. And at RGD, we do disease annotations with the RDO, which is a disease ontology that is an axiomatized and extended version of the human disease ontology, which is DO. So we've had some collaboration with DO and MGI some years ago to produce an axiomatized version which takes into account anatomy across the board, and it tends to give more than one parent for certain disease children terms. So the ontologies that I'll be concerned with today are the ones marked in by the stars on the right side of the list. So MP, VT, CMO, HPO, and RDO. We've mapped EFO to all of these different ontologies. That one, okay, sorry, I was using that one. All right, thank you. All right, so essentially the process was twofold. First, we did an automated matching of terms that our developers put together, and it was a use of the text of the term and text of the synonyms, the ID of the term, and the associated xrefs. And so all of those were taken into account for the EFO term, and then it was mapped to all the other ontologies here based on the same parameters: the text of the term, terms and synonyms, the IDs of the terms, and the associated xrefs. So the EFO was matched with CMO, HPO, MP, RDO/DO, and VT. This is the manual version of the match. Maybe 30% of most of those automated matches was covered. 30% of the EFO terms were covered by that automated matching, but then the other 70% of most of those ontologies had to be done manually. So we took into account, again, the text of the EFO term and the target term, definitions of the EFO and the target term, and the synonyms of the EFO and the target term. And we had to decide if there was a term in the targeted ontology to match the one that we got from EFO. It could be a narrow synonym, a exact synonym, a broad synonym, or something that wouldn't necessarily be a text match, but only an XREF, maybe for the term that was not quite close enough to make a synonym. Mostly that came up in matching to VT, because VT terms are rather specific as far as the disease portion of the ontology goes, and they're not much of a match for disease ontology or EFO or RDO. This is a list of the records or the terms that were matched. We started with about 7,500 unique EFO terms from the imported GWAS records, and a second batch of terms, about 2,000 worth, were hybrid terms where EFO had imported HP or Mondo terms to replace or substitute for the EFO term. And so EFO is sort of a hybrid type of ontology if you consider all across their different types of terms. So for RDO-DO, we mapped about 2,000 EFO terms. HPO, it was about 500 terms. About the same number of terms with to CMO, around 200 terms to MP. And we're still in the process of mapping to VT, which is mostly XREFs, not so much synonyms, but we're about halfway through. So we have 900 matches now, and we expect to get maybe 700, 800 more matches. Okay, now what it means for the end user of the database is a number of things, and this will give you a few examples. First, the search for an EFO term or a similar term in the other ontologies that it's mapped to will give multiple results, and they will all essentially match the disease term that was put in there, but it won't exactly it won't pick out one ontology. It will give you everything that matches. Secondly, we made transitive annotations from the human QTL pages, which are those SNPs that came from GWAS. For the transitive annotations, we took the EFO annotation that was already assigned to that SNP from the catalog, and we added HPO annotations and RDO annotations and VT annotations. So that was adding annotations to the SNP from the association of the different ontologies. And lastly, if you want to see any of if you're interested in any of the association of the ontology matches, we have a SSOM file on RGD download page, and you can see all of the matches that were made. If you're not familiar with the SSOM standard, it was a standard that was created for ontology matching, and it was published a few years ago in a paper by Matt Zolu et al. And if you're interested in ontology matching, you should check that out. So thanks to all the members of RDD who helped with all that work.
Thank you very much, Sam. We have still time for one question.
Yeah, thanks, Dan. It's clearly a huge amount of work that went into making these mappings. I guess is it something that you're thinking about or that you're already working on, which is to go back to each of the developing groups, the groups that developed each of these resources, and to share these mappings or otherwise share them through really highly visible places where those connections are widely available.
We haven't contacted anybody except VT, and they're making all the changes. They're making all the additions of the XREFs and the synonyms for us. And most of the other ontologies we have control over at RGD, so there's no one else to deal with. Your question would be, ultimately, have we worked with MP or HP? But no, we haven't. Not yet.
Thanks a lot. Let's think of a second time.